Welcome back. Today we're picking up with our second part of Chapter 6, and we left off discussing different types of mutations, and we're going to pick up talking about mutations. So induced mutations are going to be a small scale mutation. Sometimes these are often referred to as point mutations because it only occurs at a singular point. So it results when one or more nucleotide base sequences has changed, and that affects the overall amino acid sequence. And so two different types could happen. We could have what's called a substitution mutation. That occurs when a single nucleotide base has been substituted for another. So instead of, let's say you had an A there, you could have a substitute for a G or a C there. Three different types of these. The first one for the substitution can be referred to as a silent mutation. That doesn't affect the amino acid sequence at all because when you look at the amino acid sequence chart, you are able to follow through, and lots of these codons will code for the same amino acid. So a silent mutation, yes, it did change at one singular point, but it's not going to change the amino acid that it codes for. So it's still going to make the same amino acid, which would then not change that protein or enzyme. The second type is called a mensis mutation. That's where the amino acid sequence affected produces a non-functional protein. So instead of being functional anymore, now it's non-functional, so it does not do its job. And then the third is called a nonsense mutation. This substitution results in a stop codon. And so that terminates that translation of the gene that's being copied, and it produces a non-functional protein. So here is an actual visual of some of these. So you can see that you have a silent mutation, which has no effect on the amino acid sequence, so therefore it does not affect the protein. And so in this case, we had, hold on, let me get my pencil. We had this nice A here. Instead, it got substituted, and this time it changed to a T. That still coded for the same amino acid, valine, and so therefore that protein is still functional and it's still the same. The mensense is going to result in that amino acid substitution and changeover. And so with this one, you will see that the C here got substituted and changed to an A. And so it was proline, and now it's been changed over to a different amino acid that's going to change the protein, and now the protein is going to be non-functional. Next is your nonsense change. And so here at this one here, we had the C got changed to a G. In that case, it actually changed from tyrosine to a stop codon, and therefore it stops and terminates that sequence, so it doesn't continue to produce that polypeptide chain or that protein chain. And now it's not only non-functional, but it stops it from continuing to read that codon sequence as it goes through. Induced mutations are going to be small-scale mutations, kind of like point mutations, and you can have two different types of these. One's called a frameshift mutation, and that's going to occur when the DNA nucleotides are either going to be inserted or deleted, and when you do that, you completely shift how it's read. So the mRNA codons are going to be shifted into a new sequence that produces a non-functional protein. These actually can be more harmful than substitutions because it affects the overall sequence of amino acids. So it's not just hurting it in one spot. It could go in and insert it or delete it in that one spot, but it completely shifts every single amino acid from that point on, which changes how it's read and makes it completely non-functional. Let me see. Oh, sorry. Biotechnology is going to be the field of study that's going to focus on modifying biological molecules. It could modify cells, or it can modify the actual organism itself. And this has some very practical benefits. Some examples of this would be taking bacteria, inserting the human gene for insulin to produce insulin, and then having the bacteria, such as E. coli, produce lots and lots of the human version of insulin. In that case, they can take it, they can bottle it, and they can sell it to diabetics and help to keep these people alive. Before this, they would actually take insulin from cadavers, or they would take insulin from animals. Now the cadavers, it matched up, but it was hard to get. You had to get it from people who had passed. With the animals, lots of times people would have some allergic reactions to it. They would get sick, or they would have some uncomfortable symptoms and side effects from that. And this way, with the human version, you don't have that. And so it matches up with the human body perfectly. So with this genetic engineering, that's an example of biotechnology. So that involves producing genes of interest by means of manipulating an organism's genetic material through the insertion or deletion of genes. Now, it's important that can help researchers understand how genes work. We can improve agriculture, for example. We can make plants drought resistant. We can make plants pest resistant using these different types of genes. And we can develop better treatments for diseases using this type of genetic engineering. When we take a gene from one organism and insert it into another organism, that's called a transgenic organism at that point, or genetically modified organism. So sometimes when you go to the grocery store, you might pick up let's say a bag of chips, or you might pick up some snacks, and you see sometimes the GMO project on the side of that wrapper or that packaging, and that'll tell you if it's been a genetically modified organism or if it hasn't. So lots of labels, wrappers, packaging will tell you if it's been genetically modified or not. And so that simply means that they used a gene from one organism, inserted it into another organism, and now that organism is producing whatever that gene is. And so that is produced through genetic engineering. So biotechnology is the field where we can modify these molecules, cells, or organisms. Genetic engineering is going to be an example of using that biotechnology to produce something. And then that organism is called a transgenic organism. So know those different terms. Now, for biotechnology and genetic engineering, we're going to have 5 steps that have to be used for this DNA manipulation. And so that was discovered by the scientists that you see here, Paul Berg in the 1960s. So the first step you got to do is you have to cut. This is going to require the use of restriction enzymes. So we find the specific gene we want in the DNA sequence. We're going to use restriction enzymes, which are going to go in, and they're going to cut this gene out of the DNA sequence. So this enzyme was discovered by using bacteria to protect themselves from invading organisms. So what happens is these bacteria and even viruses are going to be able to go in, they can cut, and then they can destroy that invader's DNA and restrict its growth. So looking at this in the 1960s, Paul Berg realized, hey, we could possibly use this when we're studying genes that we want to start using. Maybe we can use this to cut different genes out that we might want to manipulate and we might want to use. So also, it's going to recognize that enzymes could locate a specific DNA sequence, and it can cut across the DNA molecule at the location without damaging the gene, which is going to be very important because we need to make sure we get the entire gene in order for it to be functional. And you don't want to cut anywhere like in the middle. You don't want to cut off too early or start too late with that. So you want to cut the exact sequence. So we cut. It uses those restriction enzymes. It's going to go in and recognize a short, specific sequence of DNA called a restriction site. And it's going to cut that DNA strand to produce restriction fragments. So restriction fragments are going to have a single strand ended called the sticky end. And the restriction fragments are going to produce and recognize each other, and they're going to be able to complementary base pair along those sticky ends. And so that's going to help them to form that double-stranded DNA molecule that's going to be needed. So you see in red here, that restriction enzyme goes in. It's going to cut along between that GC, the AT, the TA, and the CG. So it cuts right there, opens it up, and it makes sticky ends on both sides. And so with that sticky end, or on that one side, sorry, you have the restriction fragment on one side, but the sticky end. So it's going to come in and it's going to bond with those nucleotides that are floating around, and it's going to come in and make those complementary bases there. After you're able to do this, then you're going to go to the second step, which is called the amplification step. So we amplify. We're going to use a process known as polymerase chain reaction, or PCR, that was developed in 1985 by American biochemist Kary Mullis. This is going to help to quickly produce unlimited numbers of copies of that desirable DNA sequence. Now that's important because if you're just working with one, you need to be very careful. But if you can make many, many copies, then you can use those copies and you have unlimited sources of those genes. So that process is going to begin in a test tube where small amounts of DNA are going to be mixed with DNA primers and DNA polymerases. Remember, polymerase is going to be an enzyme. DNA is then going to be placed within the PCR machine and exposed to numerous heating and cooling cycles. So what this is going to do is it's going to kind of open it up with the heating, and it allows it to break apart and then wind it back up. So heating is going to cause the DNA to separate. It unzips, it opens up. While the cooling allows the DNA polymerase to base pair these free nucleotides so they can come in and they can match up, and then they can make more of those DNA sequences and make it double-stranded again. After about 30 minutes, you're going to have billions of copies that are going to be produced from a single DNA molecule. So that's a lot. Results, DNA strands can be used for forensics. It can be used to identify genetic disorders, or it can be used in making transgenic organisms. So it's going to be quite sensitive to contamination though, which can produce false positives. So you've got to be very careful when you're using this technology. So here's a visual. We're coming in. We are going to open up with heating. So you start out with your double-stranded DNA, you heat it, it separates the two strands. Once you separate them, then you're going to be able to see this come in, and it is going to base pair. It's going to make these strands, remember, in reverse directions. You're going to cool it, they're going to come back together, and then you do this over and over again to make billions of copies that you can eat and use. Okay, so once the DNA has been amplified, it has to be analyzed. And this is where you're going to use a process called gel electrophoresis. This is going to utilize an agarose gel that consists of shallow wells where DNA fragments are going to be located. And so the gel is going to be placed within a chamber with a positive and negative electrode, which provides an electrical current. So the DNA fragments are going to be put into each one of these wells up top. So we have this up here, and it's going to be put in there with a dye or with something that can be picked up, let's say, even by a black light sometimes. So it gets put into each one of these sample wells. You apply the electricity from the electrodes, and they're going to separate and move towards the positive electrode in, because DNA is going to be negatively charged, and so it goes towards its opposite. With the smaller fragments moving a lot faster, and larger fragments staying behind. So you got your larger fragments staying here towards the end, and the smaller fragments move very quickly towards the bottom. And so it separates these different DNA fragments out. Then they're going to appear to look like these bands here when that gel is complete. Once the DNA has been amplified and analyzed, you're going to look for those different sets that we saw there. Those are called STRs, or short tandem repeats. These are going to be short sequences of no more than 5 DNA base pairs that are repeated 10 to 15 times. It's a preferred method of DNA analysis, and it's been used to identify people's genetic profiles. It's called their DNA fingerprint because we can take it, we can look at it, and we can compare it, for example, with crime scenes. Go back. So looking at this here, we got sample A, sample B, sample C. You can go in and you can see which one matches up the best. And so let's say that this would be what we found at the crime scene. And these are our different suspects. So we go in and we look for the one that looks like it matches up the most. So let's see, we have this one here matching, this one here matching, and this one here matching for these. Now going to the other one, we have this one matching up, this one matching up, got this one, and this one down here. And so you can look to see which ones match the most, and the ones that match up, usually a lot of them will match up kind of perfectly through. That would be the suspect or the person that did it. Okay, so once that DNA has been amplified and analyzed, we got those short tandem repeats, and that's what we just looked at. So now we're off to the third step. That's inserting. This is going to involve inserting DNA into a host cell, which is known as recombinant DNA technology, or rDNA. The introduction of the desired DNA sequence into the host cell is going to require a vector. That's typically going to be small circular pieces of DNA called plasmids that are obtained from bacterial cells. Now, plasmids are first discovered by researchers studying antibiotic resistance in E. coli in the 1970s. They contain small numbers of genes not part of the bacterial chromosome. Usually anywhere from 3 to 7 genes total are going to be found in these plasmids. Most common plasmids are going to be used to extracted from E. coli because they're easily grown and they can make multiple publications exist about their genetics. So we can go in, we find out multiple things about them. And with an E. coli cell, it's going to divide about every 20 minutes. So it goes from 1 to 2, 2 to 4, and so forth. Bacterial plasmids is going to be cut using that restriction enzyme, and then we take that gene that we wanted, that desired DNA sequence, and we're going to insert it into that plasma. And then we're going to seal it up using DNA ligase, which is another enzyme. So new plasmids with recombinant DNA is then going to be inserted into its bacterial host, although viruses and multicellular host also could be used. And it gets inserted into things such as gametes or fertilized ovums. They can also be used for this. Bring this down so you can see it better. It's not going to come down anymore. All right, so here is what it is. We have our foreign DNA. We have our plasmid. We're going to go in. We cut at the restriction sites. We are going to bring the sections of DNA in that we want, attach them into those sticky ends. Then we seal them up. Now we have what's referred to as recombinant DNA plasmids, and they're ready to be inserted into their host, which is going into the main organism or is going to be grown from there. So that leads us to step 4, the growth phase. That involves the growth of the bacterial cell that contains a new plasmid with that recombinant DNA. So your bacterial cell is going to undergo a series of repeated cell divisions to produce clones of itself, which are cells that are genetically identical. Gene cloning is going to be the process in which the bacterial cell divides and recombinant DNA replicates to produce millions of copies of that desired DNA sequence. With gene cloning, it's going to be important because it can produce several copies of a desired gene that can be used in a variety of applications. Like I discussed, we can make human growth hormone or we can make human insulin using this process. So here we have a bacterium that does not take up a plasmid. It's not ampicillin resistant. Ampicillin is going to be an antibiotic. Then you have a bacterium. It's going to take up that recombinant DNA plasmid that interacts with that LAC-Z gene. So the bacterium, as it takes it up, it can't produce the galactase enzyme. And so you see these growing here. So the blue colonies are going to have non-recombinant plasmids, while the white colonies are going to have the recombinant plasmids. So that's going to be important because the plasmids are then going to be combined with different cultures of actively growing bacteria. Some cells, like we said, don't take up the plasmids. Others do take up the recombinant DNA plasmids. And then with that, your cultures are going to be cultured on a plate of ampicillin. And so the ones that actually live were the ones that became ampicillin resistant. Okay, number 5, we are going to identify. So that's our 5th step. We have to identify the desired DNA sequence that we wanted. So what you're going to do is you're going to insert or introduce a radioactive DNA probe. So a DNA probe is going to be a short single-stranded sequence of radioactive nucleotides that contain the same DNA sequence as the desired DNA strand. So upon identifying the desired DNA sequence in the bacterial host cell, the sequence will glow. So it lights up, kind of fluorescent, underneath a black light. That DNA probe is going to have also been used to identify the STRs after they've been separated by gel electrophoresis. So remember, that's your short tandem repeats. And although not typically necessary because STRs are going to be labeled during PCR, so you don't have to do this, but you can see them that way. And then you go in and you're going to identify that desired DNA sequence using dideoxy sequencing or the sugar method. So a form of DNA sequencing method is what this is. It has become more common, faster, and cheaper to use. It was introduced by a British biochemist, Frederick Sanger, in 1977. And what it does is it uses a DNA primer, DNA polymerase, and a chain terminating deoxynucleotides to identify the DNA sequence. So that deoxynucleotides are going to be modified versions of the 4 basic DNA nucleotides, fluorescently labeled so you can see them under a black light, and responsible for terminating DNA elongation. So as it's making that strand, once it hits it, it will terminate and then that strand will be done. It's going to involve adding DNA primers to the regions of interest, which begins to extend as the DNA polymerase is matching those complementary base pairs together with that primer until the enzyme reaches that deoxynucleotide. Once it hits that, the DNA fragments are going to be identified by their fluorescent dye. It's separated by that gel electrophoresis, and then it's going to be analyzed by computer software to produce a pool of DNA fragments. That pool of DNA fragments is often referred to as a gene library. And so here is what it looks like. You have your dyed sections that are labeled, and then it produces this visual that you can see and identify them. So the newer generation of DNA sequencing are going to be being developed right now that improves the efficiency and cost of doing this. You could do sequencing by synthesis. That involves a process in which each base is electronically monitored as a constructed DNA strand is making its complementary bases, so it monitors it as it's making it. We have a 3rd generation sequencing technology, and that's going to identify each base on the DNA strand as it moves through the membranous pore and passes through the electrical current. With biotechnology, it's going to be really important in agriculture. With this, we have modification of plants and animals. It helps to increase the agricultural productivity and lowers the overall cost. So genetically engineered crops are going to be first introduced to the United States in the 1990s, and since there are going to be 10 different types. And with this, a lot of people, they're not sure about it. They don't really want genetically modified organisms because you change the original sequence of that organism. And so a lot of people worry, okay, if you change the sequence, could it change how our bodies react to it? Is it why we're having more allergies, for example, or what some people are wondering? Is it why we're having larger instances of stomach cancers and colon cancers? We don't know. So for the most part, they do feel that genetically modified organisms are completely harmless. We just use them to make things, like we said, such as produce bigger, better produce. We can make it drought resistant. We can make it pest resistant, things like that. But does that change how our bodies react to that as food? It's still to be determined. Okay, so with genetically modified things that we have a lot of right now, we have corn. Most corn, about 89 to 90% of corn is going to be genetically modified. Cotton and soybeans are going to be highly genetically modified for almost all of those that are out there. So these are the most prominent and have been modified for increased herbicide tolerance and insect resistance. So that's another one I haven't mentioned is the herbicide tolerance. So they can go in and they can spray all of these fields, crop dust these fields with herbicides that will kill plants. Well, you don't want to kill the plant you're growing. So you can insert a gene to make it herbicide resistant. So only the things around it die and that plant still is able to continue to grow. Herbicide-tolerant crops are going to be modified with genes that code for an enzyme that can function in the presence of that herbicide. So that's how it's going to work. Herbicides are going to be chemicals, like I said, that are going to be used to eliminate unwanted plants by affecting their ability to synthesize or make amino acids and functional proteins. The advantage of the herbicide-tolerant crops is that it's going to allow farmers to eliminate weeds, which compete with the crops for things like water, light, and nutrients. 94% of domestic soybean acres, 98% of cotton acres, and 90% of corn acres planted in 2019 use this herbicide-tolerant seeds. Insect-resistant crops have been modified with a gene that's going to be found in bacterium called the Bt gene. The Bt gene is going to code for a protein that produces a crystal-containing spore that's going to be poisonous to insects but has no effect on humans. So the spores are going to affect the insect's digestive system by causing the gut wall paralysis and prevents them from feeding and results in death. Also, it can cause holes in the gut wall as well. So most common genetically modified insect resistance crops are going to be corn and cotton. And that's referred to as BT corn if you see questions about BT corn. Right, so here is going to be an agricultural growth chart or table. It shows the growth and adoption of genetically engineered crops in the US. And so you can see some of the different crops that you see. You see that BT corn in pink there and BT cotton. Some crops that we have that are genetically modified are going to be called stacked varieties because they contain genetic modifications that allow for herbicide tolerance and insect resistance. So in 2019, about 80% of corn and 89% of cotton acres planted used these stacked seeds that contained both of these genes. Now heading to medical biotechnology. I've already hit on this a little bit, but biotechnology has also been used to help improve the medical field with the production of pharmaceutical and diagnostic products. In 1982, the first genetically produced protein was recombinant insulin, and I told you about that one. Genetically modified plants and animals have been used to produce important vaccines, antibodies, and medicines, and essential proteins to be used against a variety of diseases and disorders. So the use of biotechnology in the medical field has also been used as a diagnostic tool for inherited disorders. Some commonly inherited disorders like sickle cell anemia and cystic fibrosis can be easily diagnosed now using biotechnology. And we have what's called gene therapy. That's going to be a process that's used to help treat and cure different types of genetic disorders by isolating their defective gene and replacing it with a functional gene. Now, the problem with gene therapy is that it's not very effective. Sometimes it gets taken up by cells that don't need it, or it doesn't get taken up by enough cells to produce enough change. So here we are, we have our first genetically produced protein, which was insulin in 1982. It's hard to believe that we did not have human insulin that we could produce on mass production levels until the early 1980s. Before that, like I said, they had to get it from animals, or there were different animal versions, or you got it from cadavers. Now they're able to insert this gene into a plasmid, put it back into the bacteria, and they're able to grow large amounts of this insulin, send it through a purification process, and then it's ready for distribution. Some of the most modern uses of biotechnology is going to be CRISPR-Cas9. And so here we have what's known as a genome editing process that was first produced by a Spanish molecular biologist, Francisco Mojica. That's going to be your image that you see on the left, and that was in the early 1990s. In 2012, we have an American biochemist, Jennifer Douana, and a French biochemist, Emmanuel Charpentier, and they're going to be the first to produce, or first to propose its use for alternation or editing of genetic material to study gene function or to correct genetic mutations. So what is the CRISPR-Cas9? Well, the Cas9 is going to be a protein that's produced by a bacteria to protect itself from viruses. Cas9 is going to be like a restriction enzyme in that it can cut specific sequences of DNA. However, instead of recognizing the sequence, it is directed to the sequence by a type of RNA called guide RNA, or gRNA. gRNA is produced from the CRISPR region of the bacterial genome. So a combination of the Cas9 protein and the gRNA forms a complex that's going to be responsible for cutting the desired DNA sequence that complements the gRNA sequence. So Cas9 cuts the desired DNA strand, which initiates the DNA repair system, resulting in an altered DNA sequence and a non-functional gene that can be studied. So it's been modified to help fix defective genes that can lead to genetic disorders. A functional gene is going to be combined with that Cas9, and the protein is going to cut out defective genes, and with the use of the repair enzyme, the DNA strand is going to be edited using the functional gene as a template. So we go in, we take out the bad gene, and we put in a good functional gene. One of the first uses of CRISPR technology was to treat a genetic disorder that occurred in 2018 when it was used to correct a defective gene causing sickle cell anemia. Hopeful that other genetic disorders such as Parkinson's or Alzheimer's or cancers can be treated using CRISPR-Cas9. They're working on those now. They're researching them. And there's going to be some concerns, though, that with this, it could affect the non-targeted genes or it could be misapplied beyond its purpose. So you can go in, you can take out common genes, that's what they're worried about, and you can put in the best of the best genes. So you can almost go in and create a designer person or a genetically almost superior person who might not be might not get some of these diseases or genetic disorders or cancers. So here we see how CRISPR-Cas9 works. We go in, you unwind the DNA, you have your targeted DNA sequence. That Cas9 enzyme, as it's reading it, will be able to take that out and replace it with that functional gene. With what we just talked about, being able to go in and fix things and change things around, that gets us into our ethical uses of biotechnology. So despite successes, using biotechnology has raised some concerns, such as safety. We talked about that with the food. Some studies shown that transgenic organisms pose no risk to humans, but many major concerns with the use of transgenic organisms include things like increased risks, like I said, with allergies. Just because you're changing that DNA sequence of the food, could it be what's affecting us? Is that why now we have so many more allergies presenting themselves than they used to? Effect on the health of the environment. Development of resistance to herbicide-tolerant or insect-resistant crops. Lack of testing and regulation. These aren't really tested and regulated a lot. And what are the hidden costs that we don't know about yet? So that's going to finish us up for our Unit 6 or Chapter 6. I'll see you back with Chapter 7.